Good day everyone, my name is Eshtiaghi. I'm an undergraduate student at the University of Manitoba, and today I will be talking about the unfolded protein response as a risk factor for alveolar rhabdomyosarcoma, also known as ARMS. What is alveolar rhabdomyosarcoma, also known as ARMS? It is known to be the most malignant childhood and young adult muscle cancer. It is called alveolar because it resembles the alveoli of the lungs, as you can see in this sample image. We invested in this research because ARMS is very malignant and highly metastatic in children and young adults, and chemotherapy has been the only available treatment option for it. Our investigations showed that unfolded protein response, also known as UPR, is highly upregulated in ARMS cells. Therefore, we went on to test the hypothesis if UPR is in fact correlated with ARMS in our human patients. What is UPR? It is the endoplasmic reticulum's response to a stressful stimuli that is caused by the accumulation of misfolded proteins. UPR mechanism has three arms: the PERK, the ATF6, and the IRE1. The focus of this research was to examine the IRE1 and XPP1 axis, where XPP1 gets spliced, sent to the nucleus to signal the proliferation and metastasis of ARMS tumor cells. The method used for this research was immunohistochemistry, where the sample cell lines were stained with antibodies specific for the proteins of our choice. The stained samples were then given to a pathologist to be scored based on their protein expression levels. They were given a score of 0 if they had no expression, 1 for weak, 2 for medium, and 3 for strong expression levels. All the data were expressed as a number percentage and compared using a time 2 test or Fisher's exact test with the SPSS software. All the p-values were presented as two-tailed, with the p-values being less than 0.05, considered to indicate a difference of statistical significance. We examined IRE1 expression intensity in our samples, and based on what you can see in this graph, normal skeletal muscles had no expression, while ARMS samples had weak and moderate expression of IRE1. Based on the p-value here as well, we understood that IRE1 expression is highly correlated with ARMS. We also examined XPP1 expression, which was also none for our control samples, while it was being weak, moderate, and strong for our ARMS samples. Based on the p-value in this table as well, we understood that XPP1 is highly correlated with ARMS and with the metastasis of ARMS tumor cells. Results of this research revealed high levels of BIP, IRE1, and XPP1 expression in the ARMS samples, as opposed to the normal skeletal muscle samples. We realized that IRE1 is highly active in ARMS cells and that it facilitates the growth of those cells. We also know that PERK would signal the cell to stop growing and undergo cell death. So we believe that inhibition of IRE1 and activating PERK would lead to a new and more effective treatment. Therefore, these proteins could be potential targets for future cancer therapy development for ARMS. I would like to give thanks to the CRIM and UCRP organization, and I would like to thank Dr. Said Rabami, Ariana Shariadi, Bhavya Bhushan, and my other colleagues who invested their resources in this research. Thank you.